मॉड्यूल में हम बात करेंगे एग कलेक्शन के हवाले से, दैट हाउ वी कैन प्रोसीड फर्दर टू जनरेट द ट्रांसजेनिक माइस, and for that purpose the first step would be collection of the eggs from the donor female. एडवांस में हमें इसकी कुछ प्रिपरेशन करने की ज़रूरत है. जितने भी reagents की बात की, मीडिया की बात की, reagents and supplies में we have to prepare them in advance. We need to prepare the DNA for injection in advance, that was cloned or any sample which you want to inject and create the transgenic mice. For that purpose, we need to purify the DNA after restriction, digestion, and after linearizing, if any, plasmid through the electrophoresis and electroillusion. Then purification is done using phenol chloroform method. Extraction is done and purification is done. Total dry this DNA and then resuspend the DNA in the DNA injection buffer at a concentration of 1 microliter per milliliter. ये जो concentration है और ये जो buffer है, that is mandatory, otherwise we may have some problem with the DNA. So we need to dilute or we need to prepare our DNA into the DNA injection buffer, and before using it we need to centrifuge it so as if there any kind of contaminating particles we can get rid of them. Now we are going to talk about steps which are required to proceed further for transgenic mice production. First one is going to be about the egg collection. For egg collection we need to prepare the female mouse that can start the ovulation. For that purpose we are going to time ovulate the female, and for that particular process we need the mare's serum to be injected intraperitoneally. 5 units are injected intraperitoneally and 100 microliter in volume is injected. अब इस इंजेक्शन का एक टाइम है that we need to observe, that is 12 hours before the midpoint of the dark cycle. You have decided which is the dark cycle, at which time point is the dark cycle, and at which time point is the light cycle. So this serum should be injected 12 hours before the midpoint of the dark cycle, and let these animals be there free for 48 hours in the cages. After 48 hours we need to inject 5 units of human chorionic gonadotropin. Again 100 microliter is injected, and following the injection we need to house this female with one breeding male overnight. And next day what we are going to do, we need to select those females which have a vaginal plug within 3 hours of the onset of light cycle. Vaginal plug means the female mated with the male and the ovulation process is started. These female mouse, they are sacrificed 16 hours after the midpoint of previous dark cycle. Midpoint of previous dark cycle, within the 16 hours we need to sacrifice them. We need to remove their oviducts in Whitten's medium in a sterile culture dish, and then that particular sterile culture dish and medium should be at the room temperature or should be acclimatized with the temperature of the working area or of the room. And we need to transfer those ovaries which are removed from those females to Whitten's medium in a second dish after washing with the Whitten's medium. Now we have to open up the ampullae of the ovaries using the watchmaker forceps. The watchmaker forceps, they have very pointed ends, and we can open up the ampullae by simple teasing motion, and we can take out the fertilized eggs or follicles that contains the fertilized eggs as well as the embryos, which are surrounded by cumulus covering. These cumulus covering and embryos, they are placed in a 5 centimeter plate in the Whitten's medium. That Whitten's medium contains the hyaluronidase enzyme. This hyaluronidase enzyme is responsible to degenerate the cumulus, and it takes about 5 to 10 minutes. Cumulus degeneration is mandatory because once the cumulus is degenerated, all the eggs or fertilized eggs or the embryos, they are released in that Whitten's medium, and we can pick and choose those eggs or the fertilized eggs. We can monitor this cumulus degeneration under the dissecting microscope, and once the degeneration process is complete, we need to wait for another 5 minutes so that the eggs can lose their stiffness. Once this process of collection of eggs is completed, then we can proceed further for the transfer of eggs using the modified pasture pipette, just like a dropper which picks up these eggs, and we can proceed further for the transfer of these eggs into for the process of micro injection of the DNA.